Welcome back to Principles of fMRI. In this module, we're going to talk about experimental design. So let's first start with a bird's eye view. Your goal is to induce the psychological state that you're interested in studying in the participant. Then, given that you've done that, a second goal is to effectively and efficiently detect those signals. What you get to control is what you present to the subject and when you present it. There are two kinds of considerations then. The first consideration is psychological. How do you get the subject to do or experience what you want them to? And then the second consideration is statistical. Once you've done that, how do you efficiently detect the results? One starting place for design is to think about blocked and event-related designs. Block design is one in which similar events are grouped together, and this was the starting point for early fMRI and PET designs, and it's still used widely today. So in one example, let's say we're interested in studying famous and non-famous faces. We might present a whole block of famous faces every couple of seconds, followed by a block of non-famous faces every couple of seconds, and alternate. In an event-related design, events are mixed together. You can see here, as we present events across time, sometimes I've got a non-famous face that's a green line, sometimes a famous face that's a red line, and they're randomly intermixed. I've even got some spacing between them called jitter. We'll talk more about that later and about how that's helpful in terms of statistical efficiency when we talk in part 2 about statistical considerations. One rule of thumb is a two-condition block design is optimal for power, all other things being equal. And moreover, a block design where the blocks are about 16 to 20 seconds long turns out to be statistically optimal. We'll go into this in more detail later, but let's start with that as a rule of thumb for now. What I want to talk about here is why that simple rule of thumb isn't always applicable. If we go back to our goals, the first goal is to induce the subject to do the thing you want them to do in the scanner. Those are psychological considerations. So let's talk about some psychological considerations and how they interact with what kinds of experimental design, blocked, event-related, or other, that we should do. And this depends on the particular task that you're studying. So there are no fixed rules, but there are some principles that you can use to understand how they play out in your own task domain and design your own task. So you have to be the psychologist and think through this in your own area. So here are 5 psychological considerations that we'll consider. One is the effect of stimulus predictability on performance. Second, the effects of time on task, how long the person's doing the intended task. Third, we'll think about strategies. Very small changes in the task design can introduce big differences in the strategies that people use. We'll think about the temporal precision of our psychological manipulations, whether we can turn them on and off when we like them to. And finally, we'll talk about the consequences of unintended psychological activity, which shows up in the brain just like activity that you intended to induce. Let's first focus on stimulus predictability. The principle is predictability influences psychological state. This is an audience participation event in this lecture, so what I'd like you to do is this simple go, no-go task. You can tap, take a pencil, tap on the desk as you're following along. So tap as fast as you can, but if it's an X, don't tap. You'll be judged and timed based on your performance. Ready? Here we go. And tap, 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 tap. Oops, I tapped. Okay. So this is an example of a block design where I blocked the no-go events and the go events. No-go events are the X's. Let's try this again. Tap, 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 no. Tap, tap, tap. No, I tapped again. This is an event-related design where the X's, the no-go events, are intermixed. The principle here is that the predictability of that no-go stimulus determines how hard it is not to respond. So if I'm interested in that control process of inhibition, then that's critical. If I use a block design, then there's psychological and neural preparation. The first X you might have tapped, it's very hard to inhibit. The second, third, fourth, very easy. There's very little inhibitory demand. With an event-related design, in this case, there's more equal demand on inhibitory processes across all the trials, on each X trial. So that's a better design. Let's now consider time on task. The principle is that you can only image the things that subjects are doing or thinking about. Very simple. So they should be doing what you want them to do as much of the time as possible. That's called time on task. Let's go back to our famous face recognition example. This is a fast and often automatic process. So many of you might recognize this face. It's Angelina Jolie. Let's work through this example. So you do this experiment where you have experimental famous faces and non-famous faces. You might recognize these faces. There's Condoleezza Rice, Condi, Cyndi Lauper, Cindy, Kofi Annan, Kofi, Jennifer Aniston, Jenny, etc. And let's say you do an event-related design and you intermix these faces and you present each face for about 3 seconds. Sounds reasonable. Let's now look at what happened during that first 3 seconds of picture viewing. You see the face. Work by Truett, Allison, and others demonstrates that the initial recognition process, which is what we're interested in studying here, happens in about the first 250 milliseconds. So I instantly recognize this person. After that, I start to think, what movie was he in? Oh, that was a great movie. I love Batman Returns. And then you might think back to your past. This is true for me. And then finally get back on task. Okay, get ready for the next trial. This all can happen within 3 seconds. So if you think about the purple block here is the time on the process that we actually care about studying. The gray block, the time on the processes we don't care about studying. So a standard event-related design contains many extraneous processes, memory retrieval and others, and you'll get lots of activation in your task, but for the wrong reasons. It won't isolate the process of interest. So what are some solutions? Well, one is rapid presentation of the faces, perhaps with a controlled task following each face presentation. And that does 2 things. It prevents the subjects from engaging in spontaneous memory retrieval processes, etc., and it maximizes the time spent doing the process of interest. More time with that initial recognition phase. Let's move next to participant strategy. So here's our example. The principle here is that different stimulus configurations, even if they're about the same quote-unquote task, afford very different strategies. Our example is the famous Stroop task, which has thousands of publications about naming colors and various similar things. So again, audience participation. Name the color in which the word is printed as fast as you can. Blue, red, green, red, yellow, blue, yellow, red. Okay. Good job. Now do it again. Yellow, red, green, etc. Okay. Second time was harder. Why is that? Well, the first block of trials, all those were compatible trials where the color matched the print name of the word. And the second block were incompatible trials in which they didn't match. And one of the standard ways of studying cognitive control since J.R. Stroop in 1921 is to compare incompatible and compatible trials. Well, like many things, the first fMRI study to do this in about 1990 by Pardo et al. Was a block design. They blocked compatible and incompatible trials. But if the stimuli are compatible, then the task that people actually do is different. If it's compatible, you might have caught yourself actually reading the words, and that's a viable strategy. If the stimuli are incompatible, then you have to name the colors. So the block design confounds the process of interest, which is the exertion of cognitive control, with task reading versus color naming, the actual task people are doing. So again, you'll get lots of activation, but for the wrong reasons. Now let's think about the temporal precision of psychological manipulations. The principle is that what you expect subjects to do should fit with what they actually can do. And it's very appealing to use block designs or event-related designs with brief alternation between event types. But let's consider an example from the emotion world in which we're asking people to recall sad versus happy memories. Is it possible to do a block design with 20-second blocks of sad versus happy memories? You go, okay, recall a sad memory. Okay, now happy. Now sad. Now happy. Well, it's not going to work. People can't switch back and forth between emotions like that. They just can't. So here's a statistically non-optimal but psychologically feasible and turns out very productive alternative. It's a single epoch design where you have a baseline state, they're resting, you do an emotion induction, They might spend a couple of minutes experiencing, working themselves into that emotional state, and then try to recover or undo that emotion if possible. So we've used this for stress designs. It also works for drug infusion designs that are difficult to rapidly alternate and that you can't repeat very easily. In the stress case, it turns out you can't step into the same river twice. So if you do the stress induction once in a person and a second time, it won't be the same or mean the same thing the second time. And that's true of many kinds of emotional states. That's why this kind of single epoch design turns out to be a viable alternative in this case. Now for our final psychological consideration. Let's think about the effects of unintended psychological activity. And the principle here is that brain imaging can capture all kinds of mental processes, including spontaneous activity between trials. This is different than a typical reaction time or accuracy experiment where you're assuming that the reaction time reflects what's happening at the moment instead of some extraneous process. Our example now is going to be spatial attention shifting, one of my old and early loves in functional neuroimaging. So let's say we're interested in brain areas that shift attention among different locations in space. So your job here is to track the dot. You're supposed to keep your eyes on the fixation cross in the middle and then direct your attention covertly to the blue dot and follow that dot, okay? There, so now here's a dot, here's the next trial, now it shifts at location, same, that's another shift, okay. So that's an example sequence of trials. Let's compare what's happening in the brain when you're switching attention versus keeping your attention in the same place. That's the goal, and that's another kind of cognitive control process. So here's the sequence of trials that we just presented. Stay, stay, switch, stay, stay, switch, stay. And typically we would model those with events, like the red for stay and the green for switch. And our design matrix and analysis are going to be based on that sequence of events. However, once again, it turns out this is not what people actually do. So if you give people a task like this, they can only maintain their attention covertly in a lateral location for a very brief time. So subjects end up shifting their attention to the spontaneous fixation cross spontaneously. So this is the likely true sequence of what happens, including all those shifts of attention, green events, in between the trials. So what's happening here? Well, you've got mismodeling of the true response in the very brain areas that are of greatest interest. Any brain area that's responding to shifts of attention is going to show a very different profile than the one that you're assuming. Okay, so we've covered 5 psychological considerations that have to do with experimental design and that are principles that we each have to think through for ourselves. We've talked about stimulus predictability and its effects on psychological and task performance. We've talked about time on task. We've talked about strategies that people might do and how small shifts in the design parameters can afford big differences in strategy. We've talked about the temporal precision of psychological manipulations and how we have to adapt the design to what subjects can do. And we've talked about the consequences of unintended psychological activity. Again, there are no fixed rules, but there are principles that we can each use and apply in our own task domain. So that's the end of this module. In the next module, I'll talk more about kinds of experimental designs and kinds of inference. In later modules, in part 2, we'll talk in more detail about statistical considerations. And that's going to come later because it has to come after we do the stuff on experimental design, GLM, and all the mechanics of statistics. Thank you.